Good morning, students. In the last class, we have discussed about the nature and the different features of the enzymes, the factors which affects the enzyme activity. Now, today we'll discuss about the different types of enzymes, classification of enzymes. Enzymes are broadly classified into 6 groups. First is oxidoreductase. In this group, the enzymes transfer of oxygen or hydrogen atoms or electrons from one substrate to another. Example, dehydrogenase oxidases. So, oxidoreductase enzymes helps in the oxidation and reduction reactions in the cell. Second group is transferase. Transfer of a specific group from one substrate to another. This enzyme induces transfer of a specific functional group. For example, transaminase. Third is hydrolysis, that induces hydrolysis of a substrate. For example, digestive enzymes. Fourth is isomerases, change of the molecular form of substrate. Example, phosphohexoisomerase. Fifth is lyase, non-hydrolytic removal or addition of a group to a substrate. Example, decarboxylase aldolase. And the sixth one, ligase, joining of two molecules by formation of new bonds. For example, citric acid synthetase. So, in that way, 6 classes of enzymes are there. Now, see the enzyme substrate reaction. Enzymes are very much specific to act on a specific substrate. First of all, enzyme and a substrate combines together and forms a complex. Bond in substrate are weakened, enzyme substrate complex is formed, and then substrate changes into product and enzymes is released. See the reaction. Substrate and enzyme, active site is there. Active site helps the enzymes to identify or recognize the specific substrate. A complex is formed, enzyme binds substrate, enzyme releases product. In some of the cases, enzymes inhibits the enzyme activity inhibited by inhibitor. That inhibitor resembles with the substrate and binds with the active site of enzyme and inhibits the enzyme activity. Now, see the another group of biomolecule, that is nucleotides. Nucleotides are the building block of nucleic acid. Basically, nucleotide consists of nitrogenous base, pentose sugar and phosphate group. See the different types of sugars present in the nucleic acid. Deoxyribose, pentose sugar present in DNA and ribose present in RNA. Next molecule, that is nitrogenous base. There are 4 types of nitrogenous base, broadly divided into purine and pyrimidine. Purines are of 2 types, adenine and guanine, and pyrimidine is of 3 types, cytosine, uracil and thymine. Uracil present in RNA and absent in DNA, thymine present in DNA but absent in RNA. See the formation of different types of nucleoside. in DNA and RNA. DNA, as you know that is a double helical structure. Two strands run together in an anti-parallel fashion, and that is linked with the hydrogen bond. DNA is the molecule present in almost all living organism as a hereditary material. RNA is the hereditary material only in some of the viruses only, like retrovirus. See the structure of DNA. Two strands are linked between the nitrogenous base. Hydrogen bonds are formed between G and C, 3 hydrogen bonds, and A and T, 2 hydrogen bonds are there. So in that way it forms the helical structure, and that was given by Watson and Crick in the year 1953. See the detailed structure of DNA. The length of one coil of DNA, that is 3.4 nanometer or 34 angstrom. The width is 20 angstrom, and between one coil there are 10 base pairs. So between the two base pairs the distance is 3.4 angstrom. Minor groups and major groups are present. Two strands runs anti-parallel, 5 to 3 direction and 3 to 5 direction. Now see the different types of messenger RNA. There are 3 types of RNA: messenger RNA, rRNA, that is a ribosomal RNA, tRNA, that is a transfer RNA. All that 3 RNA helps in protein synthesis. Messenger RNA carry the information from DNA, for the synthesis of new protein, and that steps is known as transcription, formation of messenger RNA from DNA, and then messenger RNA starts to arrange the amino acids with the help of rRNA and tRNA to form the new protein molecules, and that step is known as translation. So DNA is double stranded, RNA is a single stranded. In DNA, thymine is present, and RNA thymine is replaced with uracil. DNA is the genetic material in almost all living organisms except some viruses. RNA helps in protein synthesis. The detail of the DNA and RNA you will study in class 12 genetics. That's all.